Hi students, last time we have studied about the structure of pollen grain. Pollen grain is the circular haploid non-motile male sex organ. Today we are studying about the development of male gametophyte. Here pollen grain is the beginning of male gametophyte. This pollen grain undergoes mitotic divisions. First, it gives rise first mitotic division and two cells, that is large vegetative cell and small generative cell. Here, this generative cell floats in the cytoplasm of pollen grains or cytoplasm of vegetative cell. In second mitotic division, only generative cell, nucleus of generative cell undergoes mitosis and it forms two cells. These two cells gives rise the male gametes after second mitotic division. These two male gametes are present in the pollen grain. That time these pollen grains are set off or migrated by the process, that is the dehiscence of anther. These are the haploid pollen grains are set at two-celled conditions, and after pollination they deposited on the stigma and in time comes out through the germ pore. At its apex, there is a presence of tube nucleus, and this tube nucleus is followed with the two male gametes, and then this whole structure it is called as the male gametophyte. And this pollen tube passes through the style on the stigma. In this way, there is a development of male gametophyte takes place. I am repeating once again. Pollen grain is the beginning of male gametophyte. Pollen grain contains hexine, intine and a cytoplasm. This pollen grain undergoes two mitotic divisions. First it gives vegetative cell and generative cell. And inside there is a presence of vegetative nucleus and generative nucleus. Then this generative nucleus is divides mitotically into two nuclei, and these two nuclei are nothing but the male gametes. They get separated, and in next stage these two are called as the male gametes. These are non-motile, and at this stage pollen grains are set off or dehisced from the anther, and it is ready for pollination. It may be entomophilous or anemophilous, that is wind pollinated or insect pollinated. And when they deposited on the stigma of the female sex organ, that is the gynecium, it starts to germinate. This in time comes out through the germ pore. At its apex there is a presence of tube nucleus, that is the vegetative nucleus at its apex, and it is followed by two male gametes. And there is a formation of a pollen tube along with male gametes. That is pollen tube along with two male gametes and tube nucleus. This whole structure that is called as the male gametophyte. This is about the development of male gametophyte. Here that is the end of male gametophyte. Then next point we are studying about the female sex organ. Female sex organ of the flower is called as the gynecium. Gynecium, it is the innermost fourth whorl of the flower. It is female reproductive organ of the flower. It mainly consists of carpels. In some flowers there are many carpels, and they are called as the multicarpellary. In case of some flowers there are single carpels called as the monocarpellary. In case of some flowers there is a presence of only two carpels. Such a flowers or carpels are called as the bicarpellary flowers. When carpels are united with each other, these are called as the syncarpus. And when carpels are free, these are called as the apocarpus. Last year we have studied in the 11th standard apocarpus and syncarpus. These carpels may contain ovules singly or many. If single ovules contains, single seeds are produced. For example, maize, mango, and in case of tomato, ladyfinger, there is a presence of many seeds. That is called as the multi-ovulate. Means ovules are present. So we are observing many seeds in tomato, brinjal, and we are observing single seed in the wheat, maize, jawar, as well as mango. That is a mono-ovulate or multi-ovulate. Here we are studying in detail about the structure of ovule. Ovule is the female sex organ of female gametophyte. Actually, female gametophyte inside, that is the embryo sac. We are first discussing about the structure of female gametophyte or structure of anatropous ovule. There are different types of the ovule. Generally, 80 to 85% plant possesses anatropous ovule. Ana means inverted. Near about 15% are arthropous. Arthro means straight. Then what is the meaning of anatropous? That is inverted. Micropylar end is towards the base. Inverted. Such ovules are called as the anatropous ovule. Now we are studying in detail about the structure of anatropous ovule. Anatropous ovule contains funicle at the base. That is the attachment to the placenta within the ovary. Just above the funicle, there is a presence of a hilum, and inside this, there is a presence of diploid megaspore mother cell. Diploid megaspore mother cell. This diploid megaspore mother cell undergoes different meiotic divisions. We are studying in the development and gives rise to the female gametophyte. Here, at the center, there is a presence of a diploid cell that is called the nucellus. And this diploid cells possesses diploid megaspore mother cells, which gives rise to the embryo sac. Here in the embryo sac, there is a presence of 3 antipodal cells towards the chancel end, 3 micropylar end called as the egg apparatus. At the center, there is a presence of egg cell, and laterally to are synergids. Synergids are the filiform that is gives direction for the entry of pollen tube. Then at the center there is a presence of secondary nucleus, and it is a diploid. Although these are haploid, secondary nucleus is diploid, and it is also called as definitive nucleus. Secondary nucleus is also called as the definitive nucleus. It's another name, definitive nucleus. And this whole structure that is called as the embryo sac or female gametophyte. How embryo sac or female gametophyte developed, will discuss after this structure of anatropous ovule. Then this embryo sac is surrounded by the nucellus, and this whole structure is surrounded by 2 coverings that is called as the integuments, outer integument and inner integument. And just at its tips, there is a presence of a small space or small opening that is called as the micropyle. And micropyle is for the entry of pollen tube after fertilization. And the base of this ovule is called as the chalice. This is about the structure of anatropous ovule. I am again repeating this structure of anatropous ovule. Anatropous ovule contains funicle, that is stalk of this ovule, which is attached to the placenta in the ovary. Just above the funicle there is a presence of hilum, and inside this ovule there is a presence of the embryo surrounded by the nucellus. Nucellus, it is the mass of diploid cells, that is the megaspore mother cells. These undergoes meiotic divisions and form the embryo sac. This embryo sac is nothing but the female gametophyte. How it is developed, we will discuss after this. This embryo sac contains 3 haploid cells towards the callosal end, that is called as the antipodal cells. 3 towards the micropilar end, called as the egg apparatus. At the center, egg cell, laterally 2 synergid cells. These synergid cells are filiform apparatus and they use way for the entry of pollen tube after fertilization. And it is surrounded by nucellus. Nucellus is surrounded by 2 integuments or 2 coverings which are protective in function, outer integument and inner integuments. And here at its apex leaves a small space of opening, that is called the micropyle. Micropyle is useful for entry of pollen tube. It is a small aperture. In this way, we have completed about the structure of anatropous ovule. Then next point we are studying, that is development of female gametophyte. How female gametophyte is developed. Here we will study development of female gametophyte. That is inside the flower, inside the young ovule, there is a presence of a diploid megaspore mother cell. That is the diploid megaspore mother cell. It is present in the nucellus. This diploid megaspore mother cell undergoes 3 meiotic divisions. This undergoes meiotic divisions, that is a meiosis, and forms a linear tetrad. That is the 4 haploid cells are produced. Out of that 4 haploid cells, upper 3 degenerates or abort, and lower one remains functional. This lower functional undergoes 3 mitotic divisions. First mitotic divisions, it gives rise only, that is the division of nucleus. 2 nuclei are produced. Then second mitotic division gives rise 4 nuclei, and third mitotic divisions gives rise the 4 towards the callosal end and 4 towards the micropilar end. That is here first mitotic division. Here it is the second mitotic division, and here it is the third mitotic division. That is, there are 8 nuclei produced in a single functional one megaspore, and this matures and gives rise such a type of embryo sac. What happens here? In this embryo sac, out of that 4, 3 migrated towards the callosal end and gives rise the antipodal cells. Here it is used as the antipodal cells. Then from lower group or from the micropilar end, 3 comes at the center and gives rise the egg apparatus. It gives rise the egg apparatus. And one from each pole migrates at the center, fuse with each another and form the, that is, secondary nucleus, or also called as the definitive nucleus. It is called a secondary nucleus. And in this way there is a formation of the embryo sac takes place. That is embryo sac, or also called a female gametophyte. Here is the We are studying about the development of female gametophyte. In this way, formation of the female gametophyte. This embryocyte is also called as the female gametophyte. I am repeating once again, development of the female gametophyte. In young ovule, there is a presence of the diploid megaspore mother cell. This diploid megaspore mother cell undergoes meiosis, and which gives rise to the linear tetrad. Tetrad is 4 cells produced in a straight line, linear. Out of that, upper 3 degenerates and lower 1 remains functional ones. Or upper 3 abort or disintegrates and only 1 functional. This functional cell undergoes thrice mitotic division. Thrice means 3 times. After first time, only nucleus is divided and this cell increases in size. This undergoes first mitotic division, gives rise to 2 nuclei. Then these 2 nuclei, after second mitotic divisions, gives rise to 4 nuclei. 2 towards the chalazal end, 2 towards the micropylar ends. And after third mitotic divisions, towards each pole or polar region migrates. These migrate towards the chalazal end 4, these migrate towards the micropylar end 4, and finally out of that 4, 3 comes together towards the chalazal end and there is a formation of antipodal cells. Then 3 towards the micropylar end and forms the egg apparatus. Egg apparatus consists of large central egg cell, 2 lateral synergid cells, and centrally 1 from each pole comes at the center, fuse with each other and forms the diploid secondary nucleus, or also called as the definitive nucleus. It is diploid. These are the haploid. Means here it is 6 haploid cells and 1 diploid cell is present, and it is called as the 6 nucleated, 6 cell and 7 nucleated cells. That is the embryo sac or female gametophyte. Here we are already studying about the female gametophyte, also called as the embryo sac. And after pollination, here pollen tube of the pollen grain enters from the micropyle, touches to the synergids. It is a piliform. It breaks and it releases 2 male gametes, which we have studied just in the development of male gamete, and then 1 fuses with the secondary nucleus from the triploid primary endosperm nucleus, 1 fuses the egg cell and from the diploid zygote, and fertilization is completed. In this way, here we have completed about the development of male gametophyte, structure of anapropous ovule, and development of female gametophyte. We'll stop here. Next time continue about the pollination, definition of pollination, agencies of pollinations, and types of pollination. And after that, we will continue about the process of fertilization. Okay, thank you.